Welcome back to the GMP and Quality Fundamentals series, and this is Lecture 14. Today we are tackling one of the most commonly confused topics in analytical chemistry and GMP. Method validation versus method verification. These two terms are used every single day in quality control labs and regulatory submissions. And yet many experienced professionals are not fully clear on the difference. By the end of this lecture you will be. Let us get started. Here is what we will cover today. Number 1, you will understand the fundamental difference between validation and verification. One clear rule that makes everything else fall into place. Number 2, you will know when to validate and when to verify. This decision depends entirely on where the method came from. Number 3, we will go through all 8 key validation parameters defined by ICHQ2, R1. Specificity, linearity, accuracy, precision, LOD, LOQ, range, and robustness. Number 4, you will understand the ICHQ2, R1, guideline and which parameters regulators require for each method type. And number 5, you will learn how to apply the correct approach depending on whether you are working with a compendial method or one developed in your own lab. Let us start with the fundamental question. What is the actual difference? Here is the single most important rule. If the method is new or significantly modified, you validate it. If the method already exists and has been proven elsewhere, you verify it. Think of it this way. Validation proves that a brand new method is fit for its intended purpose. You are building the case from scratch. No one has done this before for this specific method in this specific context. Verification proves that your laboratory can successfully run a method that has already been proven somewhere else. The method works. The question is only whether your lab, your equipment, your analysts, and your reagents can reproduce what has already been demonstrated. A practical example of each. If your company develops a new HPLC method to test a product that has never been tested this way before, you validate that method fully. If your QC lab wants to adopt a USP dissolution method that is published in the Pharmacopoeia, you do not need to prove that the method works. The Pharmacopoeia already proved that. You only need to verify that your lab can reproduce the published performance. That is the entire logic behind the distinction. The method source determines which activity is required. Now let us go deeper on validation specifically. Because validation is the more extensive of the two activities, and understanding its parameters is essential for any analytical scientist or quality professional. ICHQ2, R1, is the international guideline that defines how analytical methods should be validated in the pharmaceutical industry. It was originally published in 1994 and has been the reference ever since. Know this guideline. You will see it referenced in every regulatory submission, inspection report, and method validation protocol you encounter. ICHQ2, R1, defines 8 key parameters that you may need to demonstrate during validation. I say may, because not every parameter is required for every type of method. We will look at that in the next slide. For now, let us understand what each one means. Specificity is your starting point. This asks whether the method measures only what it is supposed to measure. Can it distinguish your target analyte from interferences, degradation products, matrix components, or other substances that might be present? Without specificity, nothing else in your validation means anything. Linearity proves that the signal your instrument produces responds proportionally to the concentration of the analyte across your working range. If you double the concentration, the signal should double. You prove this by measuring multiple concentration levels and demonstrating that the relationship is linear. Accuracy is about how close your measured result is to the true value. You typically demonstrate this by analyzing samples with a known concentration, such as reference standards or spike samples, and calculating percentage recovery. If you are measuring 98% of what is actually there, your accuracy is excellent. Precision covers 2 levels. Repeatability is the precision when the same analyst runs the same samples on the same day, on the same instrument. Reproducibility, sometimes called intermediate precision, is the precision when different analysts, different days, or different instruments are involved. Both matter in a GMP environment. LOD is the limit of detection. This is the lowest concentration of analyte that can be detected, but not necessarily quantified reliably. It is the point where you can say something is present, but you cannot say exactly how much. LOQ is the limit of quantification. This is the lowest concentration that can be reliably measured with acceptable accuracy and precision. LOQ is always higher than LOD. Below the LOQ you can detect but not measure. Above the LOQ you can measure with confidence. Range defines the interval between the upper and lower concentration levels where the method has been shown to perform with acceptable accuracy, precision, and linearity. It is essentially the boundaries of your validated working range. And finally, robustness. This is about stability. How does the method perform when small deliberate changes are made to the conditions? If you change the pH slightly, change the column from one lot to another, change the temperature by a few degrees, change the flow rate marginally, does the method still give acceptable results? A robust method is one that can handle the normal variation of a real laboratory environment. Now a critical practical point. You do not always have to demonstrate all 8 parameters. ICHQ2 R1 tells you exactly which parameters apply based on the type of method you are validating. Looking at this table, there are 4 method types: identification methods, impurity limit tests, quantitative impurity tests, and assay or potency methods. Specificity is required for all 4. That is non-negotiable regardless of what the method does. Linearity, accuracy, precision, and range are required for quantitative work. Assay methods and quantitative impurity methods need all of these. But a simple identification test that only tells you yes or no does not need linearity or precision in the same way. LOD is specifically required for impurity limit tests, where the key question is whether an impurity is present above a threshold level. LOQ is required for quantitative impurity methods, where you need to measure exactly how much of an impurity is present. Robustness appears across all method types, which reflects its importance in ensuring the method can function reliably across the normal variation of lab conditions. When you write your validation protocol, you open this table and you justify which parameters you are demonstrating based on your method type. An inspector will check that your justification is sound. Now let us turn to verification. The principle is simple. The method has already been proven. Can your lab run it? This applies in 2 main situations. First, when you adopt a compendial method from the United States Pharmacopoeia, the European Pharmacopoeia, or the Japanese Pharmacopoeia. Second, when a validated method is transferred from one site to another within the same company or from a contract lab. In both cases, the method itself has been validated. The validation evidence already exists. Your job is not to repeat that work. Your job is to prove that your specific laboratory can reproduce the published performance. Here is how verification works in practice. Step 1. You select the parameters to verify. For most verification exercises, you will focus on specificity, repeatability, precision, accuracy, and the limit of quantification if the method involves trace analysis. You do not run all 8 validation parameters. You focus on the ones that are most likely to vary between labs and equipment. Step 2. You run the tests. You prepare your samples using your reagents and your equipment. Your analysts run the method under your conditions. You generate your own data. Step 3. You compare your results to the acceptance criteria. For a compendial method, the pharmacopoeia or the originating site defines the criteria. For a transferred method, the sending site defines them in the technology transfer protocol. Your data must meet those criteria. If it does, the verification passes. Step 4. You document everything. This is where many labs cut corners and it causes problems during inspections. A verification report is a regulatory document. It must contain the protocol that was approved before you started, the raw data, the calculations, the results compared to the acceptance criteria, and a clear pass or fail conclusion. The report must be reviewed and approved by quality assurance before the method is used for GMP testing. No exceptions. This decision diagram summarizes the entire lecture in one image. The question at the top is, where did the method come from? If the method was developed in your lab or significantly modified from an existing method, the path leads to full validation. You go through ICHQ2, R1, you demonstrate all the relevant parameters, you document the results in a validation report, and you get QA approval before using the method. If the method came from a compendial source like USP or EP, or it was transferred from another validated site, the path leads to verification only. You confirm the key parameters, you compare your results to the acceptance criteria, you document everything in a verification report, and you get QA approval. The critical point in the middle is this: you cannot skip either activity. Some labs assume that because a method is in the pharmacopoeia it needs no demonstration at all. That is incorrect. Verification is still required. It is less work than full validation, but it is mandatory. Let me now highlight the four most common mistakes I see in practice. The first is using a compendial method without verification. This is surprisingly common. A lab receives a USP method, assumes it is approved because it is in the pharmacopoeia, and starts using it without any verification. This is a finding in every regulatory inspection where it occurs. The fact that the pharmacopoeia validated the method does not mean your lab can run it. You must demonstrate that. The second mistake is treating verification like a mini validation. Verification is not a shorter version of validation. It is a different type of activity with a different purpose. Scope it correctly from the beginning. Check which parameters need to be confirmed and run only those. The third mistake is validating once and never revisiting. Method validation has a life cycle. If you change the HPLC column brand, the water system, the instrument model, or if the product matrix changes, those changes may affect the method performance. A change control assessment should determine whether partial or full revalidation is needed. And the fourth mistake is not defining acceptance criteria before running experiments. Your validation or verification protocol must state the acceptance criteria before any data is generated. Defining them after you have seen the results is a data integrity violation. Predefined criteria are a fundamental GMP requirement. Let us bring it all together. 6 things to remember from this lecture. Number 1. Validation proves a new or significantly modified method is fit for purpose. You build the evidence from scratch. Number 2. Verification proves your lab can reproduce an already proven method. It is applied to compendial methods and transferred methods. Number 3. ICHQ2, R1, defines the 8 key validation parameters: specificity, linearity, accuracy, precision, LOD, LOQ, range, and robustness. Number 4. Not all parameters are required for every method type. ICHQ2, R1, gives you a clear decision matrix based on what the method does. Number 5. Both validation and verification require documented protocols and reports with predefined acceptance criteria. These are regulatory documents. And number 6. Method validation has a life cycle. Equipment changes, matrix changes, and process modifications may trigger revalidation or re-verification. Your change control system should catch these triggers. That is Lecture 14 complete. Coming next in Lecture 15, we go into method lifecycle management. What happens to a method after validation? How do you maintain it, monitor its performance, transfer it to another site, and eventually retire it? This is the full picture of how analytical methods are managed throughout their life in a GMP environment. If this lecture helped you, please give it a like. Subscribe if you are not already, and leave your questions in the comments below. I reply to everyone. I will see you in Lecture 15.